All right, welcome back. Today we will talk about learning and memory. This will sort of serve as a transition from when we used to talk more about nuts and bolts, neuroanatomy, action potential synapses, and things like that, and into how do these actually create behaviors? What are sort of the higher functions? A lot of times for students, particularly those with a more psychology background, this becomes when the class gets more interesting. So as an overview, we're just going to really start basic, introduce a few terms, some jargon, some fundamentals of learning and memory. And then in sort of thinking about it, we've learned a lot about the synapse, and we're going to transition that knowledge into learning about learning and memory. So we're talking about some work that was done just to understand sort of real basic understanding of how learning and memory works at the level of synapse from the C-slug aplasia. And similarly, we'll talk about long-term potentiation, LTP, another molecular event that happens at the synapse that is important for learning and memory. So, real basic questions. I don't have a huge love for definitions, but I think they are important for us to be able to talk about things. So, in class, I would have asked you these things but think for a second, what is a memory and what does it mean to learn? And how are those two processes different? Right. So what I would say is that memory is sort of a product of learning and experience, and learning is a relatively permanent change that results in behavior from interactions with the environment. Or another thing to think is I think a learning is sort of something change behavior due to experience over time, if that makes sense. Whereas memory is a product of learning, so something more of a functional unit. Now, we don't really always understand what that unit is, but I think that helps us to understand it. And we'll come back to it. There's different types of learning and memory, and I think that's why it's hard to really define it with one definition. Right. So when we talk about learning and memory, I think it's real critical that we sort of go back to the basics of how we first started to think about that. And the most obvious one, I believe, is sort of Pavlov's dogs, right? Where Pavlov had these dogs and he would feed them every day. And as is natural, before you're going to eat, we do this too, you start to salivate. And the dogs would do this naturally. Now, Pavlov noticed every day before he came in, he would ring a bell or make some sort of noise. The different books say something slightly different. And he started to realize that the dogs were salivating in response to that bell rather than the meat itself. And so he developed this sort of way of thinking about this where, these all grouped into unconditional and conditioned responses and stimuli. So the basic idea is you don't have to learn this, right? Unconditioned stimulus, in this case, is food. And the unconditioned response is the salivating. Right? That's unlearned. No, it just happens that way, right? However, the conditioned stimulus was that bell that was made noise, right? That is something that the dog was learning to respond to. And then, the conditioned response, and this is usually a little bit tricky, is the salivating response to the bell. So often the unconditioned response and the conditioned response are the same thing, but it's in the context of which they happen. Are they happening to a conditioned stimulus or an unconditioned stimulus? This I'm going to mention now. In the future, it'll be a few classes around the next research paper you read. Understanding conditioned and unconditioned stimuli will be very important for understanding the paper. But the main point to see here is if we come back to our definition, they learn to respond. So it's not as though the first time you play a bell, they salivated, right? This took multiple times over and over again. And that's the process of learning, a changed behavior over time in response to environment. This is more what will be used in your paper that you're going to read. And this is an example of eye blink conditioning, whereby this is a rabbit. That's what's normally used for these. And if you, just like us, if you had a little puff of air into the eye, that's what this tube is here, you would blink. And now what you can do on top of that is, and what they're showing up here, is you can play a little tone for the rabbit right before they're going to get the puff. And eventually they'll start to blink in response to the tone to sort of protect their eye. And this is something that we can use to understand how they're learning. And do they start to learn that with fewer trials? We could say, or that being better at learning. Right? So you can see here, conditioned stimuli for a certain amount of time, and then you get an unconditioned stimuli right at the end. And you can see that as they're learning, the conditioned response increases over time. Does that make sense? I don't think I explained that super well. The idea is, in this figure, showing how this is sort of a schematic of how you might learn, right? So you have the normal conditioned stimulus, and then you have the unconditioned stimulus, CS and US. Right? And what it's trying to show here is that over time, the conditioned response happens in response to the unconditioned stimulus. As you read the paper, I think this will make more sense. All right, so what types of memory are there? So the first type of memory we think about usually is called declarative or explicit memory. This is our ability to recall facts. What did you eat yesterday? What does your grandmother look like? Things like this are sort of explicit or declarative memory. And these are strongly rooted in the hippocampus and the amygdala as well. I think sometimes people say the amygdala is sort of also with it, and some people say it's sort of equally important. It seems to depend a little bit on the type of memory. Fear memories, smell memories seem to amygdala is extremely important. Still trying to understand how it was the other one. So this is the basic type of memory that people talk about. We won't focus on it a ton, but I want you to be familiar with other types of memory because they're important as well. And they tend to broadly fall into this category of implicit memory. So things, influence of experience on behavior, right? If that's sort of our general definition. A lot of these can talk about contextual learning, weather prediction, right? You don't have to, like if you walk outside and you're like, oh, it feels like rain today. That's not like you had a factual memory recall of like, oh, this exact temperature with this pressure and this sort of cloud, oh, it must rain today. No, you just had a sort of instinctual feeling that falls into this category. Same with the classical conditioning we just talked about and things called probabilistic learning, which we do an awful lot of. Just sort of, if you played sports, you start to, you generally be familiar with this, you know what to expect just because you've seen something repeatedly. All of these tend to be largely sort of the basal ganglia. The most common type, I feel like, or the one I think of the most for implicit memory is always procedural memory, and that is your ability to sort of learn a physical skill. You know, riding a bicycle, you know, learning to a golf swing or a baseball swing, different things like this where you don't actively recall them, but you do them and your body is just able to. And these, we really strongly think of the cerebellum playing a strong role. Right. Another term that I'm going to come to, and one of the videos you're going to watch, this will be talked about even more, is the Ingram. And the Ingram, I feel, is like what connects neuroscience to psychology and sort of really delineates how we would approach learning and memory a bit differently. And neuroscientists are very interested. There's a search for the Ingram is something we're all searching for. And this is physically, what is a memory in your brain? And we have better understanding. That video that you'll watch, the TED Talk, will talk about that quite a bit. But we still don't exactly know what is the physical representation or physical thing in your brain that a memory is. And you know, this is very much what a neuroscientist is looking for. What we do know, though, is that to make a memory, something in your brain must physically change. And what that is exactly, it's usually something of the synapse or structurally about neurons making more synapses that lead to that. All right. So a lot of students learning memory, they're very excited to learn about it. But I think I always try and give a caveat there. We don't understand well how learning and memory works in humans. We talked about, I mentioned this, right? The hippocampus is important, and we know that from things like patient HM, which we'll hopefully talk about later. And then we know the cerebellum is important for procedural memory. But at the level of molecules and neurons, we don't have a good understanding. But where we know this extremely well is in the sea slug aplasia. And you're going to watch a video on this as well. Dr. Eric Kandel won a Nobel Prize understanding how a very specific memory event works in the sea slug. And he understood it very well. That's the key, is that he took this simple animal with a very simple, almost reflex, and understood it very deeply in a way that you couldn't with a more complex animal. This whole approach, and generally the approach of using sort of animals, is called reductionism, right? We want to start small and try and understand it very deeply and then apply that to higher organisms. And much of what he found in his lab found in the sea slug turned out to be true in much more advanced animals and probably ourselves as well. So the key here is that the sea slug, is what they're trying to show here, has a set of gills which it needs to protect because that's really important, right? Sea slug, if you don't have gills, you die, right? But they also need to be open to the water to let water come in and filter through it, right? So what the sea slug does is it normally leaves it open, but then if you touch, and that's what's showing here with the probe here, it withdraws the gills and protects them with a layer of skin, basically. And what they realized was, and this is sort of a classical conditioning event, is that if you touch this repeatedly, eventually it learns, oh, nothing's going to happen. So I can just leave the gills out there and nothing's going to come and eat me or eat, right? I don't need to protect these gills. So how does it do that? That's really all the question they were trying. So how does it learn not to withdraw its gills after so much time? This is literally the most basic circuit you can imagine, right? So there's the siphon, right? This is really that mantle that was on there, the part of the skin that is outside of there. And then there's a sensory neuron that can sense if it's been poked, and it connects to a motor neuron that's connected to the gill and then can pull it in. So basically how this normally works is sensory neuron's active, that activates the motor neuron, withdraws the gills, and they're protected, right? Repeatedly activating the sensory neuron, eventually these synapses between them become weaker. So then the motor is no longer able to activate the motor neuron unless you touch this with a much more times or with a stronger stimuli, right? That's learning. The simplest learning event I could probably think of, right? This used to respond, then it realized that it no longer needs to, so it stops. So what is happening here is somehow changing these synapses that connect this one sensory neuron to this one motor neuron. Now, that last one was the main take-home here, and this is sort of a scary looking figure. I don't want you to spend too much time here, but just know that they were able to also understand there's a difference between short-term and long-term memory. And the main idea is that short-term works very locally, just at one synapse even, and it sort of is slight alterations usually to the neurotransmitter receptors or different ion channels that can either strengthen or weaken that, letting more ions in or out. Usually, if you've taken more biology, a sort of phosphorylation event comes that alters these ion channels. And that can be reversed. So that usually we're talking about last hours, maybe a couple days, maybe usually less than 24 hours. So long term, we're talking about a change that affects usually the whole neuron. And you have to go back to the nucleus and change basically what genes are being expressed, what genes are turned on and off in response to this. And this changes lots of synapses, and it usually changes the number and size and overall shape of the neuron and synapses. So a much more global change for long term, a very local change for short term. All right. Last thing, and I think I will just put this in this one video, though feel free to take a break here before we dive into long-term potentiation. All right. Long-term potentiation, which everybody just calls LTP, and I will too because it's kind of a mouthful, is a very important event at the synapse that we know is key for learning and memory. So basic idea here is that persistent firing between neurons can lead to enhanced ability to induce changes in memory potential. Basically what this means is that if one neuron is very actively firing and the next neuron is responding to that, that leads to a stronger synapse where you can get even more response to the first neuron. This can be short, a few hours, minutes to hours, or it can be long-term days. So that satisfies what we need there as well. Right, and this is again a question I would have asked there, but I hope this is obvious. The short and long-term versions of LTP, that should make sense because we have short and long-term memories as well, and we did in the C-slug as well. So you can see where this molecular event could be a useful strategy to use for learning and memory as well. So here's the main idea of a real basic figure showing it. We're basically, you can see here, they're looking at the hippocampus, and this is a slice culture. You don't have to worry about this, but they basically sliced off a chunk of the hippocampus. They put a stimulating electrode in one part and a recording electrode in another part, like we've talked about in class. In the dish, you can imagine this with one cell. Now we've got this much bigger section of tissue, but it works the same. And normally, what they're trying to show here is if you stimulate, right, you get some response that is a little bit 0.02. So this is a normal low-level stimulus. You stimulate over here, and then you record over here, and the recording is saying somewhere, a little below 0.02. Don't worry about what the units on there. It's kind of a weird thing to think about for right now. Then, if you stimulate like crazy, which is what it's trying to show here, you induce LTP, where now, if you go back to normal stimulating, you can see that you're getting a much higher response, over double. And this starts to slowly decay, but it takes a while, right? We're now, nearly an hour out, and we still see an elevated response. And if you had done an even stronger response here, this would have lasted for longer as well. Right? So that's the basic idea of what LTP is. Now, how does LTP work? This is, I feel like, the deepest molecular thing I'll end up going over in the class, but I hope you'll understand a few points here. So, this is usually at a glutamate synapse, right? So this axon up here is the presynaptic neuron, their axon. It's releasing glutamate onto a synapse. That synapse has two types of glutamate receptors. The AMPA receptor, which is a glutamate receptor, and the NMDA glutamate receptor. Usually, the NMDA receptor doesn't do much of anything because it's got this magnesium molecule that's blocking it right here. However, and then the AMPA receptor does the majority of what's happening there. Right? Now, the thing is that the AMPA receptor usually only allows a small amount of sodium in, whereas the NMDA receptor, if you could activate it, allows more sodium in and also can allow calcium in. So that is going to have a much bigger effect on the change in voltage of the other cell. It's going to really create a big rise. So what happens now, though, is that if you release a whole bunch of glutamate over a long period of time, that lets a bunch of sodium in through the AMPA receptor, and that creates a sort of high voltage consistently there that it kind of kicks out that magnesium molecule that was blocking the NMDA receptor. So now the NMDA receptor can work as well. So this creates this huge now change in voltage in response to the same amount of glutamate that was being released. In addition, this is normally what we think of sort of short-term LTP. If you do this for a long time, you start having additional synapses here now that can respond as well. So it's showing here an additional AMPA receptor that now creates an even greater depolarization in response to the glutamate, because now you have more receptors, more synapses, and you have a much stronger link between those two neurons. All right? Okay, so this is a paper I've always found pretty interesting. It's getting kind of old now that I look at the citation, 1999, but I think it's a cool way to see it, how we might understand how this has a practical effect on learning and memory. So if we look over here, this is the same graph we saw before. There's black triangles and white squares. And if we activate them, they're showing the same response. However, here we cause LTP, and now you start to see the white squares are behaving differently than the black. I guess there's black squares and hand triangles. So what's the difference here? Well, so these come from two different lines of mice. The white ones here are from wild type mice, just normal everyday life mice. The black ones here have a particular subunit or part of that NMDA glutamate receptor that's been changed in a way that it's actually more like ours. Kind of interesting, right? It's this NR2B, for those people who want to know, and they've put in the special version of this that seems to be important for learning memory in us. And what they see is that by doing that, they get a much higher and longer lasting long-term potentiation. So that's great. That works in the ways we think about it for physiology. But I think is really interesting that this goes all the way to change their behavior. So everything from here on out, the rest of the figures are showing white is those wild type normal control mice. Black are transgenic mice that have this special glutamate receptor part. And basically, the transgenic mice seem to learn faster, which this was sort of the paper that proved that this has a real physiological effect. So let's walk through these two different assays because I think they're important to know. So this is a contextual freezing assay. When a mouse gets scared, it freezes, or when it doesn't know what to do. So basically, the idea is if you put something in a cage with a mouse that should scare it, you would expect it to freeze. And so they show here, and this is showing additional trials doing it. So the first time you do it, that's completely new. Both mice freeze. The mouse, the sort of special mouse, seems to even freeze a little bit more. But right away, by the second trial, that second mouse doesn't freeze as much because what it learned was that even though that was new and unknown, it's not actually scary. It didn't hurt it. It didn't do anything bad to it. And you can see that over time, it learns much faster than the wild type mouse that takes, maybe sort of 4 trials to get what the transgenic mouse learned in the second trial. And then this is a very similar assay as well. What I also want to spend more time here, though, is on this, right? This is called a Morris water maze. And the idea is, and I might try and pull out and the idea is, if I can draw up here, right? Let's see here. Yeah. So if you put a mouse in a pool that's like this, right? And you put a little platform here, and it learns that the platform's there. So you just put it in there a bunch of times, it learns that it's there. And the mouse, you drop in the water, it's going to want to swim to that platform because mice just don't like being in water that much, right? They don't want to drown, basically. So then what you can do is you can divide this into quadrants, right? And what you expect is a mouse that has learned is going to get there pretty quick. But a mouse that doesn't remember where it was will kind of swim all the way around and eventually get there. So it'll spend more time in other spots. So that's what it's showing down here, is that mouse, the wild type mouse, is kind of looking all over. It spends a little more time in the target one, the one with the platform, but in the other ones, it spends almost as much time. Whereas the special mouse spends a lot more time in that target because it's really sure. It remembered that was there, so let's just go right there to it, right? Let's see that for now. Yeah. So, what other changes might result in LTP? Normally, I'd ask you to talk about this more, but I'm just going to give you one of them. If we're talking about changing in synapses, I don't know if you remember that on dendrites they have these little spines, right? And this is where synapses were. And this was another sort of fundamental paper from the field that basically showed as LTP is induced, and that's what's showing down here, that you get the formation of these spines and they tend to get larger. So it's the first time we could see there's a physical change at the end of these dendrites that actually changes, and that causes LTP, or facilitates LTP. And this is, if you can imagine that we had a microscope and you could peer in here and you saw one of these rooms, that could be a sort of engram, right? That could be a physical thing that's happening that causes learning. This could be a memory that formed here in response to this. I guess here is better. So again, many mechanisms of LTP. Just remember, like we talked about with the aplasia, the sort of early LTP, the short term, that's going to happen very locally at single synapses. Long-term late LTP is happening across the whole neuron, and it's usually making new synapses, changing the shape of the dendritic spine, changing the shape of the neuron, increasing the number of neurotransmitter receptors and the types of them. It's having a much bigger change, and that's why it results in longer, stronger, lasting connections. All right, so I'm just going to save this for now. We have lots of open questions on how memory works. I feel, you can look at this two ways. Some students are always disappointed, I didn't learn exactly how it works, but I hope that you look at it the other way, as though there's a lot that we have left to understand, and we have a good foundation to understand that moving forward. But if you have any questions, please contact me, and I'll try and work them into the classes. If you have very specific or more specific questions in particular, I can try and work those into future classes. All right. So next class, which we'll have a Zoom session for, I'm going to follow up on LTP because I know that's a little bit tricky one to get. We'll do some discussion questions about memory that we would normally do in a regular class, and we'll talk about spatial memory, which was sort of a recent Nobel Prize, about how we understand where we are and how we're moving in relations to everything. That is what I have for this video, so thanks for watching.